Welcome to lesson 2.6.2, the cell cycle. This can also be found in your textbook section 10.2. What we're talking about today is trying to find out what are the main events of the cell cycle, and really we can break them down into three main events. The first one is as anything that is new and it is living, a cell needs to grow. So the first part of the cell cycle, the cell is going to grow. Then the cell is going to start to prepare for division. So the cell prepares for division. It's going to copy the things it needs to copy. It's going to build the things it needs to build in order for the cell to divide. And then finally, the cell is going to divide. It's going to reproduce. So the cell divides. Alright, so in prokaryotic cells, this is a regular pattern of growth. DNA replication and cell division. It happens all the time. Most prokaryotic cells begin to replicate or copy their DNA once they have grown to a certain size. So size matters in prokaryotic cells. While the DNA replication is when the DNA replication is complete, the cell divides through a process known as binary fission. Binary fission is an asexual form of reproduction. And what that means is you don't need a mother and a father, you just need one cell to reproduce. And so this one bacterial cell, and bacteria are an example of binary fission, this one bacterial cell is going to replicate or duplicate its DNA, and then we're going to have this pinching action, which is called a cleavage furrow, by the way. We're going to have this pinching action until that pinches all the way across, and we get two brand new identical cells. So we're going to have two identical cells. All right. So this is a little bit different than in eukaryotic cells. In eukaryotic cells, we can split this system up. We can split this cycle up into two main periods or two main sections. We can split it up into interphase and we can split it, and then the second part is what we call the M phase. So let's take a look at interphase. Interphase is going to be most of the cell's life. It's going to go through three periods, G1, S, and G2. Basically, the G stands for growth, and S stands for synthesize. So that may give you some clues as to what's going to happen there. M phase really stands for cell division phase. And so cell division is going to be split up into two phases. It's going to be split up into mitosis and cytokinesis. But for now, we're just going to call it the M phase or the cell division phase. In the G1 phase, we're talking about a new cell that's just been produced, and we're going to have growth. So during G1, we're going to have cell growth. It's going to grow in size. And as it grows in size, in order to do that, we need to synthesize proteins. Synthesize proteins that will be a part of its structure. We're going to need to synthesize proteins that are worker proteins or enzymes that the cell needs. So the cell is going to produce many enzymes during the G1 phase. And it's also going to synthesize new organelles. Synthesize new organelles. Most of the cell's life is spent in the G1 phase. So this is worth most of the life. Most of cell's life. But there comes a point where we need to think about dividing in cell. Either the cell is getting too big, or there's some space for a new cell because some old cells have died off. Something has happened in which we need to start thinking about cell division. But before we do that, we need to go through the S phase. And S phase stands for synthesis. This is the synthesis phase. And what's being synthesized is new DNA. This is when DNA replication takes place. We have to copy all of our DNA prior to dividing the cell up so that each new cell gets the right amount of DNA. So we're going to replicate the DNA, and during the G1 phase, during this phase, our DNA is in the form of chromatin. So right now the DNA is in chromatin form. And it is during the G1 phase, and it will be during the G2 phase. So again, I'm putting this slide in for everyone to take another look. Remember that DNA during DNA replication needs to be unwound by helicase and needs to be held open at the replication fork by single-stranded binding proteins. Then for our leading strand, we're going to start out with an RNA primer and then built by RNA primase, and then DNA polymerase is going to build continuously following the helicase molecule. On the other side of the molecule, we have the lagging strand, which is going to be built by we're going to have an RNA primer built, and then DNA polymerase is going to build opposite the direction of helicase. And then helicase is going to open it up a little bit more, and we're going to get RNA primer, new DNA. We'll open up the DNA a little bit further, and we'll get an RNA primer, as is shown here. If I can find my pen, as is shown here, and then new DNA. So we're going, first piece, second piece, third piece, to get these Okazaki fragments. And then the Okazaki fragments are going to be put together by a DNA ligase. And then when we finish replicating the whole DNA, then telomerase is going to come in and fill in those end segments where the RNA primer once was. Once we're done replicating all of the DNA, we're going to move on to what's called the G2 phase. And remember, I said that the G kind of stands for growth, but the cell's not going to get any bigger. So the cell doesn't grow. The cell does not grow. However, what the cell is doing in terms of growth is it's making organelles that it needs for division. So it's going to produce organelles that are specifically needed for division. And the other thing that we're going to do here that really isn't about cell growth, but it's getting prepared for that cell division, is we're going to produce molecules that is needed for cell division. So this is really a preparation phase right before cell division. We're going to prepare for mitosis. Finally, in the M phase, this is cell division. And mitosis, it's split up into two things. We have mitosis and we have cytokinesis. Mitosis is the splitting of the nucleus. It is the division of the nucleus. It is not the splitting or the division of the cell. It is nuclear division. And so I want to make that distinction very clear, that mitosis, we're going to split the nucleus. We're going to split the contents of the nucleus up into two new nuclei. In cytokinesis, we are actually splitting the cell, or splitting the cytoplasm into two equal portions. And we'll talk more about these in our next video. But remember, we have G1, S, G2 phase as part of interphase, and then the M phase is split up into mitosis and cytokinesis. So during the cell cycle, a cell is going to grow, it's going to prepare for division, and then it's going to divide into two new daughter cells. I hope everyone has a good night, and we'll see everybody in class.